So welcome to the second portion of point two, where we'll continue our discussion of gastrointestinal infections and agents of food poisoning. In the previous presentation of part two, we have briefly reviewed the clinical syndromes produced by and characteristics of Campylobacter jejuni, Salmonella, both typhoidal and non-typhoidal, and Shigella species. So in this part 2.2, we will continue and conclude our review of the clinical infections produced by Vibrio, E. coli 0157H7, Listeria monocytogenes, Yersinia enterocolitica, and briefly discuss the conditions caused by Helicobacter pylori and Clostridium difficile. So we'll start with cholera. The word cholera is derived from the Greek chol, Khol, meaning bile, and rhin, Rhin, to flow. Cholera is another interesting disease, and I would like you to do a search for Jon Snow. No, not that Jon Snow from the Game of Thrones. Jon Snow, who played a major role in the discovery of the waterborne nature of cholera after observing fatalities around the Broad Street pump in Soho, London in 1854. And the study of epidemiology was born. This is just a matter of interest. No, you're not going to be tested about Jon Snow. I just want you to expand some of your background and interesting knowledge about the history of some of these diseases. I did not have time to tell the story of Typhoid Mary when I talk about typhoid fever. The story of Typhoid Mary is a classic chronic typhoid carrier, but you can easily again do a search about her as well. Very fascinating. So cholera is an extremely unpleasant and potentially fatal disease caused by Vibrio cholerae. Vibrio cholerae causes a life-threatening secretory diarrhea by stimulating the adenylate cyclase system of the gastrointestinal tract. The disease presents abruptly with watery diarrhea. Reports back in the early centuries essentially said that people, your neighbor could be alive one day and dead the next. That's how quickly this disease can overcome an individual. Fever, abdominal pain are seldom prominent, but if they occur at all, but death occurs by dehydration. Treatment requires aggressive fluid replacement, with antibiotics having a secondary role. Cholera is rare in the United States, being seen almost exclusively in travelers, but the possibility of an eighth pandemic of the disease remains real. And again, cholera vibrio 01 is the most serious of all the vibrio subtypes. Characteristics of vibrio species as a review: they are gram-negative bacilli, curved or straight. They're oxidase positive. They are fermentative and salt tolerant. That means they are halophilic. They are susceptible to 0129 vibriostatic compound. That is a hallmark of their identification to differentiate vibrio species from other oxidase positive fermentative gram-negative bacillus. Halophilic vibrio species produce various types of clinical infections. Vibrio parahemolyticus is an important cause of gastroenteritis associated with the ingestion of inadequately cooked seafood or food that has been contaminated with sea water. Raw oysters are incriminated in about 1/2 of the cases in the Gulf Coast states, where the disease is most common in the United States, but outbreaks are associated with other types of shellfish. Eating raw oysters during months ending with R, that is September to December, has been what you call an old wives' tale kind of thing, where the effect of, and also the effect of climate changes in emerging infectious diseases, such as vibrio infections, may be of a matter of interest. When we were stationed in Florida along the Gulf Coast, we almost always include TCBS agar as a highly selective agar to isolate Vibrio, as it was not uncommon for us to recover Vibrio parahemolyticus, as well as the other halophilic Vibrio species, Vibrio alginolyticus and Vibrio vulnificus, in our samples received in our laboratory in Florida. Explosive water diarrhea is usually the first symptom and is often accompanied by crampy abdominal pain when infected with Vibrio parahemolyticus. The organism can be isolated, as I mentioned, on TCBS agar. The disease is usually mild and self-limited. Vibrio vulnificus causes an infection that occurs after eating seafood, especially, again, raw or undercooked oysters, most commonly, or through open wounds when swimming or wading in infected waters, or via puncture wounds from the spines of fish, such as tilapia or stingrays. Because Vibrio vulnificus is 80 times more likely to spread into the bloodstream in people with compromised immune systems, especially those with chronic liver disease, such as those who are alcoholics. Severe symptoms include blistering skin lesions, septic shock, and even death can occur. So this is where Vibrio vulnificus is not uncommon among people with liver cirrhosis, like I mentioned, as a result of alcoholism. And when we talk about raw oysters, you eat raw oysters with beer or beer with raw oysters. They sort of go together. So that's some kind of background information that you may want to think about. While Vibrio alginolyticus, we talk about Vibrio alginolyticus associated with in wound infections contaminated with marine water. It manifests with bacteremia. It's also an uncommon source of ear infections swimming in marine water. Again, when we were stationed in Florida, we had a case of an infected wound with Vibrio alginolyticus when this man was working on his boat and his leg was injured. So again, these are the halophilic Vibrio species and the different types of clinical infections they produce. Laboratory identification: when suspected, a transport medium such as an alkaline peptone broth with sodium chloride should be used, again because they prefer salt. So inclusion of this highly selected medium, TCBS, for example, may be appropriate in certain geographic locations if you are stationed or live along the Gulf Coast, where you would most likely encounter this group of organisms. So review the characteristics of this Vibrio species when grown on TCBS selected medium. Review the growth patterns in peptone broth without sodium chloride and compare it with growth in supplemented 3 to 8% sodium chloride. That will help you in the identification and differentiation of Vibrio species. So for example, Vibrio cholerae will grow without any salt supplement. So it will grow 0% sodium chloride up to 3% sodium chloride because it's non-halophilic. While halophilic Vibrios will not grow in the absence of salt. So 0% sodium chloride in the peptone broth, and growth will be observed only in the 3% and 8% sodium chloride. Also observe the colonies on TCVS. Species that are sucrose fermenters will produce yellow colonies, while sucrose non-fermenters will show us green colonies. Use this as your initial differentiating key characteristics to differentiate the different Vibrio species. Let's move on with E. coli 0157H7 and other Shiga toxin producing E. coli. In addition to E. coli 01757, there had been other serotypes that had been involved in foodborne incidents in Shiga toxin producing E. coli. And those, some of those are listed on the right-hand side of this slide. The strains of E. coli that produce Shiga toxin, also known as enterohemorrhagic E. coli strains, or ETHEC, were recognized in the United States in 1982, and I believe it was Taco Bell started that outbreak of E. coli enterocolitis due to hamburgers served in, was it Jack in the Box? Oh, I believe it was Jack in the Box. No, I'm mistaken. It was not Taco Bell. It was Jack in the Box. And now had caused an estimated 20,000 cases of diarrheal diseases each year. Although uncommon in primary care, these strains can cause severe hemorrhagic colitis, hemolytic uremic syndrome, and thrombocytopenic purpura. Most human infections have been transmitted by beef and are thought to result from fecal contamination of meat during slaughter. Undercooked hamburger meat had been famously associated with these outbreaks. It has been estimated that a hamburger purchased at a fast food restaurant in the United States contains meat from as many as 1,000 cows. Talk about detail here, fascinating. Outbreaks have also been associated, however, with the ingestion of unchlorinated drinking water, unpasteurized apples, cider, alfalfa sprouts, lettuce, mesclun lettuce, radish sprouts, milk, and other foods and beverages. You know those fruit juices that is freshly made, but they are unpasteurized. They have been sources of E. coli contamination as well. New standards for food processing have been introduced in the United States with the aim of reducing the prevalence of this pathogen. However, the infectious dose is low, possibly as low as 100 bacteria. Therefore, prevention is difficult, and person-to-person spread may occur as such in the case of outbreaks in restaurant chains. And the latest, I believe, was the Chipotle outbreak. Love it. E. coli 0157H7 differs from most E. coli strains by its production of one or more Shiga toxins, and a key characteristic feature is its inability to ferment sorbitol. There are E. coli other than Shiga toxin producing strains, as I mentioned, the usual, such as ETEC, which is the enterotoxigenic strains, the usual cause of travelers' diarrhea. So here shows sorbitol McConkie agar is a screening agar for E. coli 0157H7 if that is suspected. E. coli, this strain does not ferment sorbitol, therefore will produce clear colorless colonies, while on the regular McConkey, it will show up just like any other E. coli. Other screening tests is something that we call MUG assay. It detects beta-glucuronidase, serological typing, and toxin detection. Most laboratories may not perform these types of assays and may be sent and will be sent to the State Health Department for confirmation when this organism is suspected, particularly in outbreaks. The next is Listeria monocytogenes. We talk about and beat this to death as far as Listeria monocytogenes. These are gram-positive non-spore-forming rods. So we'll just review Listeria as a foodborne pathogen. They're usually acquired or transmitted through previously cooked food products, foods that we think it is safe to eat as is, hot dogs, sausage, milk, cheeses, ice cream, raw vegetables, fermented raw meats, sausages, etc. I suggest that you take that into consideration and think about it as far as your own safety and your family when you're dealing with these food products. I suggest that you nuke your hot dogs like I do, and I do burn sausages. Be very careful when you're eating raw vegetables as they're easily contaminated. So no, you don't soak them in bleach, but I do use dish detergent in rinsing my vegetables. Yes, I'm fanatic about that. Again, this is nothing new. Microscopic morphology, gram-positive, non-spore forming, Coccobacillary, right? Cultural characteristics, grows well on blood agar, produce a very narrow zone of hemolysis, very similar to group B streptococci. Review the identification characteristics of Listeria monocytogenes, including that this is catalase positive. It is motile at 25 degrees, showing that umbrella type motility, but it is non-motile at 37. It also has a tumbling motility characteristic in hanging drop preparations. Yersinia enterocolitica and yersiniosis. Yersiniosis is a bacterial disease that usually affects the intestinal tract. This particular species in the United States, most human yersiniosis is caused by the species Yersinia enterocolitica. It is more frequently found in cooler climates and occurs more often in winter than in summer months. So I lived in Texas for many years, and this is one organism I have to admit I did not encounter Yersinia enterocolitica because they essentially get fried with the heat in Texas. So unlike with Vibrio, we saw quite a bit of Vibrio in Florida, did not see Yersinia at all over the years that I spent my time in Texas. Yersinia infections are acquired by eating or drinking food or water that is contaminated with feces from an infected human or animal. Animals, especially pigs, are the main source of Yersinia. Waste from infected animals may contaminate water, milk, and food and become a source of infection for people and other animals. Raw pork and pork products, especially pork intestines, are often contaminated. The bacteria have also been found in cold cuts, raw or improperly processed milk, ice cream, tofu. I roast tofu, I don't eat raw tofu. Shellfish in lakes and streams, and both in wild and domestic animals. Infection with Yersinia can cause a variety of symptoms, depending on the age of the person infected. Common symptoms in children are fever, abdominal pain, and diarrhea, which is often bloody. In older children and adults, right side abdominal pain and fever can be the predominant symptom and can be confused with appendicitis. So in some cases, the patient will present with symptoms and clinical presentation of an acute appendicitis, will be taken to surgery, and the surgeon essentially will find a very normal looking appendix. He'll take it out anyway, because we really don't know what the function of the appendix is other than have an appendectomy. Anyway, that's on the side. But what the surgeon will find in cases of eosinosis is inflamed mesenteric lymph nodes, and determine that what the patient has is a mesenteric lymphadenitis. So the symptoms can be confused with appendicitis. So that is one of the hallmark of this. Remember that one of the hallmark of eosinosis is that the presenting symptoms mimicking appendicitis, when in actuality the disease is really mesenteric lymphadenitis that is being caused by Yersinia enterocolytica. Review the characteristics of Yersinia enterocolytica, such as the use of highly selective agar, CIN. What does CIN stand for? Cifsolidine, ergosin, novovicin agar. This is a very highly selective media for isolating, recovering Yersinia enterocolytica. And the requirement for cold enrichment. And this is done by inoculating a phosphate buffer broth and incubating the inoculated broth at 4 degrees refrigerated temperature overnight before you culture it on your CIN agar. And this is supposed to enhance the recovery of Yersinia enterocolytica. Remembering that the optimal growth for this organism is between 25 to 35 degrees. So enhancing it at the cold temperature supposed to enhance the recovery of Yersinia enterocolytica. It is non-modal at 35 degrees, but modal at 25 degrees. It is weakly urease positive. Again, review the biochemical characteristics of Yersinia enterocolytica, but mainly how best to isolate this organism. And these are the key identification features. So as I mentioned, the last couple of agents that we'll talk about, and I really giving you the responsibility of reviewing more about the organisms, are Helicobacter pylori and Clostridium difficile. So with Helicobacter pylori, we don't get involved in the clinical laboratory much in isolating Helicobacter pylori, mainly because the diagnosis is done via detecting the organism's high urease activity. So although Helicobacter pylori does not cause diarrhea, it has been associated with peptic ulcer disease. We thought that peptic ulcer and gastritis was caused by stress, which kind of is, but later on discovered that it's actually it is actually an infection being caused by this organism, Helicobacter pylori. Microscopic morphology is very similar to Campylobacter when it was first discovered as a cause of peptic ulcer and gastritis. The genus, it was placed in the genus Campylobacter because of its gram stain or microscopic morphology being gram-negative spiral shaped or curved rods. But through DNA homology studies determined that it belongs to a new genus Helicobacter. Active infection can be diagnosed by detecting the organism on a gastric biopsy or through a breath testing, as I mentioned earlier. This testing is based on the organism's ability to produce urease and break down urea. So what they do essentially give the patient carbon labeled, usually carbon 13 or carbon 14 labeled urea, and when ingested will produce labeled CO2 in the patient's breath, and it's like doing a breathalyzer, and it will show a high level CO2 if the organism is present. For me, I was diagnosed with Helicobacter pylori infection through a biopsy in a routine duodenal endoscopy. So yes, actually, Helicobacter pylori is not uncommon for older the population after age 50. The incidence of Helicobacter pylori infection increases. With regards to, just to close up this portion with Clostridium difficile, review how we detect Clostridium difficile. And one thing that you want to remember with Clostridium difficile is the clinical infection that it produces is antibiotic-associated diarrhea. So the event usually occurs after a patient or individual has been placed under a long-term or prophylactic antimicrobial therapy, and a gastrointestinal event subsequently develops, which then can be diagnosed as an associated antibiotic-associated diarrhea, and you're able to relate that with Clostridium difficile, which then, if it remains undiagnosed or untreated, will become our subsequent complication is pseudomembranous colitis. So the last few slides, I just want to talk about non-bacterial food poisoning. Let's say these are more of chemical food poisoning rather than bacteria causing the gastrointestinal disease. So this is about a 32-year-old woman who ate a fish dinner at a restaurant. Before she returned home from the restaurant, she began to feel warm. She developed headache, abdominal cramping with some diarrhea, and was generally flush. Symptoms continued for a few hours, but then abated. Actually, this was my case. This was me after eating some kind of tuna tartare in the restaurant. On the way home, I started to feel ill. Okay, so just a few minutes with the agents of non-bacterial food poisoning. Chemical intoxications are often associated with fish consumption, non-bacterial food poisoning. The case presented in this in the previous slide is typical of scombroid. And this was me, as I mentioned. The syndrome, which consists of flushing, headache, and crampy abdominal pain with diarrhea, is caused by ingesting contaminated fish, often tuna, mackerel, or yellow jack. The tissues of the fish contain histamine and enzyme inhibitors, which are responsible for the symptoms. And I am very allergic to histamine. I have so many allergies, it's unbelievable. So the symptoms usually start within 1 hour of ingestion, sometimes within minutes of ingestion, and usually only last several hours. I thought it was forever. So fish and shellfish poisoning are just beginning to be recognized. So for example, this amnesic food poisoning caused by Nischia pungens. It's a diadem ingested in mussels. This is a diadem that produces domic acid, which is ingested in the viscera of mussels. Paralytic shellfish poisoning, or PSP, and ciguatera occur when high levels of toxins produced by sea algae or when dinoflagellates are ingested makes its way through the food chain, causing what we call the red tide. The red tide is a common term used for algal bloom. These are colonies of algae. These are simple plants that live in the sea or fresh water. And when they grow out of control, they produce toxic or harmful effects on people when ingested by fish and go through the food chain, especially. So when there is red tide, caution or alert, you remember or take note that eating shellfish is sort of prohibited, or decent restaurants will not sell or buy shellfish when there is an alert for red tide because of this algae. Anisakis. Anisakis infection is associated with eating contaminated sushi or sashimi. Anisakis species is, of course, a parasite. So I love sushi, but you have to know where you get your sushi, and that is a reputable sushi place. So significant clinical findings, travel history, and food intake, incubation period, and initial laboratory findings are helpful hints as far as how to proceed or where to start in the investigation of gastrointestinal infections and diarrheal events. That is how you should be able to determine your method of recovery and defining the characteristics of the intestinal pathogens on this screening media. Another point to remember would be the key identification features not only of the pathogen, but the hallmarks of each of the different infections that we covered in this lecture. This is the end of this lecture for this week.